I think I'm meeting you virtually, at least Jan, for you for the second time. I think we did one more. I remember we did one more virtual event that was with Alexander Tomoscu.
I see.
I forgot that was a yeah that I forgot the event name. That was in Georgia, I think.
Oh,
So there was a special session on pangenomics, I feel, uh, as part of a big event in Georgia that happened in Georgia.
I see.
Okay, never mind.
Well, we are happy to have you here today. And okay, so the plan is to start in 10 minutes. I think that so I will introduce you. And so, is it correct that you are now assistant professor at the Indian Institute of Science, and that have uh, you have got your PhD at Georgia Tech University, and after that you have been a postdoc at an NIH. Is that
Yes,
fine?
yes, yes, yes.
Okay. Okay. Um, Maybe it's time if you can try to share the presentation so that we can check if everything is fine. And
Yeah. Is there a recommended browser for Google Meet? I'm just wondering, like, sometimes in Safari doesn't work as well.
Simone, you're muted.
oh, sorry. I was saying since it's Google, I
Okay.
suppose Chrome, but I'm using Safari without any issue. So
Oh, okay. Okay. So maybe because as soon as I pressed the share screen button, it started
oh,
like
okay. Hmm.
it started that load like
Yeah, the loading.
let me see if I have to.
Yeah, you think it's feasible to try another browser?
Sure, let me do that.
Amazing.
Yeah, we see it.
So we are seeing your entire desktop right now.
Okay. Let me just let me see. So play window. So is that okay? Like let me see. So is this okay, or do you prefer like a full screen?
Um,
The point is, what I'm seeing is your desktop. I
yeah,
mean, there is, I don't know if it's Safari or Chrome. I don't see your presentation. You're
Okay,
showing.
so maybe I should try the sharing thing again.
Okay, now I see that.
So do you
Okay,
see
now
my
I see you
slides?
moving. Okay,
yes,
I see
yes.
the slide, but not in presentation mode.
And then when I do it,
That's
still
perfect. Yeah, it's not as large as
okay. So let
the
me.
screen. Yes, it's not fully maximized.
How about now?
Yes.
That's perfect. Yes, that's
Yeah, yeah,
perfect.
yeah.
Now you're moving through the slides, and that's amazing.
Okay, perfect. Is it okay if I go and grab water quickly?
Yeah, sure.
Oh, yes, sure. We have 6 minutes,
Okay,
but
perfect.
we can wait a couple of minutes more if needed. Don't
Okay,
worry.
thanks.
That's perfect.
Ah, Gianluca, posso chiederti un favore?
Sì.
Potresti provare a scrivere sulla chat di YouTube perché mi diceva ieri Luca che non riusciva a scriverci e non so se c'è qualche permesso da cambiare.
Allora, vediamo subito. Ci va uscire. Senza permesso. Ha ragione. Live chat, join the conversation, open panel e disabilità.
Non ho sentito.
Ha ragione, non si riesce.
Ok.
Perché io vedo, c'è live chat,
Mhm.
join the conversation, ma c'è un pulsante open panel che è disabilitato.
Mm.
Aspetta, no, però è qua, sono scemo. Aspetta, faccio test. No,
Ok, lo vedo.
sì, 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 sì. No, ok.
Uh.
And your name is Chirag Jain, correct?
Yeah, Chirag.
Okay, Jain.
Chirag Jain.
Okay, I'll try to do my best.
And how do I pronounce your name?
Gianluca,
Gianluca. Okay.
that's can be quite fine.
Could you remind me the time? Like, shall I assume 30 minutes?
Well, we have we have 45 minutes if you wish. I mean, the next speaker will be in an hour, so there is plenty of time. If 45 minutes for you is fine, that's perfect. If you have planned for a shorter talk, that's fine too.
Okay.
But we try to be sure that we have plenty of time for everyone. That's the advantage of online meetings. You don't have to pack
Yes.
everything to very short time.
Yeah, today also happens to be ISMB deadline, so I guess many people may be busy, like especially people who are submitting to ISMB very late.
Yes, yes. We are trying to make a deadline also, but I hope that our student will be able to make it. Anyway, it's
Sorry, Gianluca, just a sec.
yep.
Because I saw our next speaker trying to join, um, but she disappeared. Okay, sorry.
So I'm very glad to start this morning in our time session with Chirag Jain. Chirag is assistant professor at the Indian Institute of Science. He has published several papers in computational pangenomics, and I'm really excited to have him here. Before joining the Indian Institute of Science, he got his PhD at Georgia Tech, and he has been a postdoc at the National Institute of Health. So I see from the title that you are going to talk about the problem of genome inference in a pangenomic setting. So thanks again for joining us, and we're waiting for your talk.
Okay, thank John Luca. So, yeah, so my name is Chirag Jain. I'm a faculty member at the Indian Institute of Science. So it's a pleasure to talk to you all today about our recent work on pangenome-based genome inference via integer programming. So let me get started. And before I get started, I can mention that so my talk is not is short. So basically, that means that there is a lot of time to ask questions in the middle. So please feel free to interrupt me by directly speaking, and I'll stop and answer your question. Okay, so let's get started. So I think all of you have seen the overview of Pangenome graphs already, but just for completeness, let me remind you that the graph-based representation of pangenomes is one of the compact ways to represent a large collection of human genomes. This is this representation is made possible because of recent advances in genome assembly, the ability to get complete haplotype resolved genomes, and as a result, these graph-based representations also allow us to capture the variation in the human populations. So as a result, it addresses the limitations of the conventional reference that we were using, which was a string-based data structure. Now, a graph-based representation offers these benefits that can be helpful for improving genomics accuracy. So there are a number of applications of pangenomes, and especially in the context of human genomics, they allow us to mitigate the reference bias, uh, allow us to enhance the disease association with studies, and of course, getting insights into population genetics and evolution. For those who are still learning about this topic, there are excellent review articles written on pangenome algorithms and pangenome applications. So I refer you to those articles for more information. Now, addressing these applications is also not easy because pangenome graphs are now complex compared to the string-based representation. So there are a number of algorithmic challenges. For example, building the pangenome graphs itself is not easy. You have a very long sequences coming from large number of individuals, and determining how to construct an appropriate graph such that the common sequences are collapsed into a single haplotype or into a single vertex, and you can spell the individual haplotypes as a path in the graph. That's not an easy problem to solve. Similarly, mapping reads to a pangenome graph is another challenge because now there is a lot of combinatorial possibilities that you have in the pangenome graph. And finally, if calling variants, like even once we have the accurate read alignments, or we have the short read sequencing dataset or long read sequencing dataset, calling variants in the target sample using a Pangenome reference is also challenging. So in our group, we have been interested in all of these problems, and we have contributed some algorithms for each of these directions. For today's talk, I'm going to focus on the genotyping problem, which is our most recent work that is going to appear in Recon conference. So the next few slides, I'll give you an overview of what we did in this work. So just as a background, let me just quickly remind you what genotyping means. So in case, suppose if your target genome is haploid, that is the ploid is equal to 1, then the genotyping is simply the process of identifying if a known variant is present at a specified locus. So for example, a known variant would be SNP, insertion, deletion, or a large structural variation. The process is to identify whether that variant occurs at a specified locus or not. In the case of diploid genomes, then the genotyping problem is to identify whether a known variant is present at both chromosomal copies, or just one chromosomal copy, or none of the chromosomal copies. That is, we have to account for the fact that now the ploid is 2, so therefore the genotyping has to be done on both copies of the chromosome. So given this background, there are a number of approaches there for genotyping problem. This is a fundamental problem in genomics. So if we think of the solutions that involve the use of a linear reference, and let us say that you have sequenced the target sample using short reads, then we have two kinds of methods. That is, one is alignment based, and the second is alignment free. That is directly, for example, using K-mer counts from short reads. Now, alignment-based methods would align these reads to the reference, and based on the consistency of the alignments and how, whether it's a match or a mismatch, one would infer the genotype. And in the case of alignment-free method, you would use K-mer counts to infer the appropriate genotype. In this scenario, one can also use haplotype panels. For example, if I have some reference panels of large number of individuals available already, I can use those panels for phasing as well as imputing missing genotypes. For example, if there is a specific location where I am not able to infer the genotype with confidence, then I can also consult the reference panel, and there are statistical frameworks that would allow us to use these panels for phasing and imputation. So, but all these solutions use a linear reference genome, which has limitations, because then the limitations of linear reference is that we would face reference bias in polymorphic regions, such as the regions involved with immunogenetic regions. And also, when we have short reads, it can also be challenging to genotype large variants, especially structural variants, as well as variants in repetitive regions, because there both alignments can be inaccurate and even K-mer counts can be ambiguous for repetitive regions. So this is a rough overview of how approaches work using a linear reference. And as a result, there have been a number of attempts on extending the genotyping problem for using graph references as well. So in the graph-based reference, you have now a Pangenome graph reference, right? Instead of a linear reference. And again, I can think of two kinds of approaches, that is alignment-based and alignment-free methods. So for example, in alignment-based methods, again, I would want to align these reads accurately to these graphs. And based on the alignments, I would infer the genotype at all locus. And similarly, in alignment-free methods, based on the K-mer counts, for example, the K-mer frequency, for example, I can consider the K-mer at this path, what is the frequency of those K-mers in the short reads. Similarly, K-mer frequency of this path, K-mers, and what is the K-mer here, and what is the frequency of those K-mers in the short reads. And based on the K-mer counts, one can also infer the most likely allele that would be part of the target sample. So the key advantage of these Pangenome graphs is that both small and large variants can be represented well inside these Pangenome graphs. So as you can see as illustration, this illustration is from the Pangeny paper, where you can already see that these variants would appear as bubble-like structures in these graphs. And the problem then is to infer a path in these graphs that would match the allele of the target sample. So given this overview, basically the goal of our work was to develop a rigorous algorithmic framework for genotyping using pangenome graphs. And the motivation here is that most of the existing approaches require complex heuristics for solving this problem because graphs can be very challenging to operate with. So in this attempt to identify the rigorous algorithmic framework, we first start with the assumption that let us say that the target genome is haploid. So we start with a relatively easier problem where we assume that the target genome is haploid, that is ploidy equals 1. And now we can think of this genotyping problem as finding an optimal haplotype path in the graph. That is, let us say that's a mosaic of the known haplotypes in the graph, and it agrees with the given short reads. So we will have short read sequence from the target genome. So we would want this optimal haplotype path to agree with the short reads. I'll formally define what I mean by this word agree. And we would want this optimal haplotype path to be a mosaic of the known haplotypes. That is, we allow recombinations. That is, one can switch from some haplotype to other haplotype in the Pangenome graph. And this idea is partly motivated by the Lee-Stephens haplotype model and also the Pangeny work, which also uses a similar concept. So given this intuition, let me give you a precise problem formulation that we are working with. So let us say we have a Pangenome graph which has, let us say, some haplotypes represented as paths in the graph. So for example, you have 3 haplotypes shown here, one in dark blue, one in light blue, and one in green color. We also have reads from the target sample, and let us say from reads we have identified the k-mer matches to the graph. So for example, this substring ACCC is matching to a subpath in this graph spelled by this path AC and CC. So accordingly, you will have a number of k-mers in the short reads, and many of these k-mers would have one or more matches on this Pangenome graph. So this is the input, and the optimization problem that we are framing is as following. Given this set of k-mer matches, and let us call them as anchors, we want to determine a haplotype path in the graph with minimum cost, where I'm defining my cost as the number of recombinations, that is, how many times is the path switching between haplotypes. Plus, I would also penalize any anchor paths missed in the inferred haplotype path. That is, let us say that if my haplotype path is not consisting of this k-mer match, then I would penalize for this missing this k-mer match, because that means that my haplotype path is not fully agreeing with the given sequencing reads. So here is an example. Let us say that you have my inferred path is shown in pink color. Then this pink color path has one recombination. So there would be a cost of recombination because I'm switching from this dark blue path to light blue path at this point. And also observe that I'm missing some of the k-mer matches. So for example, this substring CCTT is not matching as a path to this inferred path anymore because I missed this anchor, right? But other anchors I have captured, because you can see that this anchor, the anchor here, and the anchor here, they have been included. But the penalty of this exploding this match would be accounted for. And as a result, the number of recombinations in this case would be 1, and the number of k-mers missed in the inferred path is also 1. So therefore, in this particular example, the cost would be 2. So given this objective function, you want to determine the optimal haplotype path, which minimizes the cost defined here. And the hope is that this optimal haplotype path would be the best representation of the target genome. So let us start with solving this problem and analyzing its computational complexity as well. So when we look at its computational complexity and to decide to see whether this problem is even, do we have a polynomial solution to this problem, or is this problem NP hard? So it turns out that this problem is NP hard, and even if we restrict to binary alphabets. We show this proof by drawing a reduction from the Hamiltonian path problem. So let us say you have a Hamiltonian path instance where you are given an instance with 4 vertices. Now I'm going to convert the instance given to the Hamiltonian path problem into an instance for the haplotype path inference problem. And this I will do by drawing a pangenome graph in which I will create multiple copies of this graph. So for example, if there are 4 vertices in this graph, I'm going to create 4 columns in this pangenome graph. And if there is an edge from U1 to U2, I'm going to put an edge from U1 to U2 across all these columns. So for example, there's an edge from U1 to U2 here, again there's an edge from U1 to U2 here, and there's an edge from U1 to U2 here. So the connectivity of these vertices will be defined based on the connectivity of the original instance. Now, the way we show the reduction is by putting the k-mer matches on this graph. And the way we put the k-mer matches is basically we also have a source and a target vertex here. We will put many number of k-mer matches on the source vertex and large number of k-mer matches on target vertex as well, so that the inferred path is forced to start from source vertex and it must end at target vertex to be optimal. Otherwise, it would lose a lot of anchor matches, which would result in large penalty. Also, we create unique string matches to each of these vertices such that the inferred path is forced to visit each vertex at least once. So for example, if I consider vertex A here, then it should go to T, then it can go to G, and then it must go to C to cover all the string matches. So as a result, such a path would only be possible if there is a Hamiltonian path in the original instance. And through this way, we draw an equivalence between the Hamiltonian path problem and the path inference problem that we are trying to solve. So we show the equivalence both ways. That is, if you have an inferred path, then the original instance has a Hamiltonian path. And similarly, if you have the original instance as a Hamiltonian path, then we will have an inferred path with a certain cost. So now through this equivalence, we know that the path inference problem is NP hard. So then what do we do? Should we give up, or should we design some kind of efficient solutions that can still work on practical instances? Well, it turns out that this problem can be solved efficiently using integer programming. And in the next few slides, I'm going to walk you through the integer programming solutions that we have developed to address this problem. And in the end, I will show you that even if the problem is NP hard, we can use these efficient integer programming solutions to scale to problem sizes that are practically relevant. So let me start with the overview of how this integer programming framework works. Okay. So as a first step, what I'm going to do is for the given pangenome graph that we have as input, I'm going to create an expanded graph where I'm going to separate all the haplotypes from each other. So for example, remember that in the pangenome graph, 2 haplotypes can share a common vertex. But in my expanded graph, I'm going to separate all these haplotypes and I'm going to introduce a source and a sink vertex such that I have separate paths for each of these haplotypes. So I'll have one path for haplotype 1, I'll have one path for haplotype 2, and I'll have a third path for haplotype 3. Also, wherever a recombination is possible, for example, at vertex CC, observe that I can switch from haplotype 1 to haplotype 3. So to account for this possibility of recombinations, I would also put these red colored edges to account for the possibility of recombinations. That is, I want to allow recombinations up between these haplotypes. Now, moving forward, what I'm going to do is basically introduce the integer programming constraints that would depend on the number of edges and number of vertices in this graph. But one unfortunate drawback of this expanded graph is that the number of recombination edges between adjacent columns can be quadratic in number. That is quadratic in the number of haplotypes. Because as you can see, maybe you may want to switch from every haplotype to every other haplotype. So in that case, you would need to introduce quadratic number of edges, which might later affect the number of constraints as well, because then we will need too many constraints in the integer programming framework that we are going to introduce. So as an optimization, what we are going to do is we will optimize this graph a little bit. And to optimize this, what I'm going to do is the recombination edges, I'm going to reduce them from quadratic to linear. And that the way I do it is I introduce a dummy vertex in the middle of these columns, so that whenever I want to do a recombination, I would go to the dummy vertex first, and from the dummy vertex, I would go to the other haplotype. And this way, the number of recombination edges between adjacent columns remains linear. Okay. So now, once we have this optimized expanded graph, I'm going to start framing my integer programming variables and constraints. So let us say that I will put also the weights on these edges. So I'm going to assume that the weight on these black colored edges is 0, because I'm not facing any recombination penalty for these edges. However, I'm going to put a positive weight on these red colored edges, because this is where I have to put a recombination penalty if my inferred path is going through these red edges. So there I would, for each red edge, I would put a weight of C by 2 if my recombination penalty is C. Because for any recombination, I'll have to take 2 consecutive red edges, which will account for recombination penalty C. So now, okay, so now I hope that this expanded graph definition is clear to you. That is, you have these separate paths for each haplotype, and you have these edges connecting the different haplotypes to account for recombination cost. The black colored edges have weight 0, because there we don't want to penalize any recombination, whereas the red colored edges have a positive weight. Okay, so now I move forward, and just to remind you, the goal is to find an inferred path where we want to minimize the number of recombinations as well as the number of anchor paths missed. So the idea is as following, that is, I'm sorry, just one second. Okay, so the idea is as following, that we will use a binary decision variable for each edge of this graph. And I'm going to convert this problem into a network flow like problem in which that binary variable will be, the value will be 1 if there's a flow of 1 or along that edge, and it will be 0 if there is no flow along that edge. And the network flow analogy comes from the fact that we will try to seek a flow of 1 from the source to the sink vertex, and along the flow, because we have weighted edges, we will check for whether the flow uses the black colored edges or whether the flow uses the red colored edges, and that will determine the number of recombinations in the inferred path. So these binary decision variables will tell us the flow going from source to sink along all the edges of this expanded graph. For example, if my inferred path starts from source to sink in this orange colored manner, then all the edge variables along this path will be 1, whereas all the other edge variables will be 0. And the intuition is that whenever we see this red colored edge, whenever we see a flow of 1 along a red colored edge, that means this is something that we have to minimize, right? Because that's the recombination cost. As well as there is a second constraint, that is we have to also account for these anchor paths missed from the short reads. And we do this by checking whether for each k-mer match, we do not penalize a k-mer match if all the edges in that subpath have a flow of 1, because that means the inferred path is including that k-mer inside it. However, if the inferred path has any edge, so if we have any edge variable which has value 0, then this will be penalized because we are missing this k-mer from the inferred path. So this is the intuition of how this integer programming framework is working. So let me show you how the exact equations look like. So we develop two type of constraints. One is the integer linear programming, in which each constraint is a linear constraint. We also develop integer quadratic programming, in which the constraints can be quadratic in terms of the variables. So let me show you the integer linear programming framework, in which we have an objective function where you can see that I'm minimizing the weight times the edge variables, because the weight is telling me the recombination cost, and the other part is for the anchor matches. So to walk you through these constraints, basically these constraints will look familiar because this is the flow conservation constraint. That is, we have to make sure that the incoming flow minus the outgoing flow should be equal to 1 only if the vertex is the source vertex. It should be -1 only if the vertex is the sink vertex. And the incoming flow should be equal to outgoing flow for all other vertices. We have other constraints for the anchor matches. That is, we want to penalize the anchors which are not part of the inferred path. And for that, we put the anchor constraints as well. And in this case, it turns out that the number of string hit constraints, that is the number of anchor hit constraints, is equal to the total number of anchors in the graph. Now, this can sometimes be too many constraints, and that's what we wanted to optimize further. So as a result, we also developed this quadratic programming framework in which we are allowed to have quadratic constraints as well. And as you can see, this is the quadratic constraint in which two variables are having a product with each other. And this time, again, the same analogy, that is, we have flow constraints, and this time the number of anchor hit constraints are reduced. But the number of anchor hit constraints are now only the number of k-mers in the reads that have a match in the graph. Whereas in the previous case, if a k-mer has, let's say, 5 matches, then we will have 5 different constraints. But now we have only a single constraint for each k-mer in the read. But as you can imagine, this is a trade-off, because now even though we have reduced the number of constraints, our constraints are more complex, because these are quadratic constraints instead of linear constraints. So we see that there's an interesting trade-off between this linear and quadratic programming frameworks, and we see that one is faster and one uses more runtime when we use Groby solutions. Finally, we further optimize this linear programming framework by relaxing these edge variables. We show that even if we relax these two continuous variables, the optimization still returns optimal answers. So this is an overview of how we optimize this integer programming framework so that it can scale to practical instances. So this is how we implemented this integer programming framework in a software called Phi. Phi stands for Pangenome-based haplotype inference. The inputs are short reads and Pangenome graph. For each haplotype, we will compute these anchors in parallel. Then to start the integer programming frameworks, we will construct the expanded graph. and we will add the constraints for on top of the expanded graph. And once we have the constraints and the variables, we will optimize the integer model with Gurobi. So finally, the Gurobi solution will return us the flow from source to sink that will be used as an inferred path in the Pangenome graph. So let us see how this method compares to existing solutions for the genotyping problem. And so let me walk you through the experiments that we did. So this is an integer programming framework. So right now it's not scalable to the entire human genome, but it scales to the specific regions of the human genome. So we used focus on the MHC region, which is the polymorphic region on chromosome 6. It is about 5 million base pair in length. And we used publicly available short reads from this MHC region of 5 homozygous human genomes. And we are using homozygous genomes because, as I said, we are still assuming that we are working with haploid genome, where we are looking for a single haplotype path in the pangenome graph. Now we use this homozygous genomes because we have the ground truth assemblies for these 5 cell lines. So using long reads, researchers have assembled these sequences, and we have complete curated sequences for these MHC region of these 5 cell lines. So then we have a ground truth to compare with. So when we infer the path and we genotype a path in the pangenome graph, then we can check we can compare it with the assembled sequence from long reads and see how well it is matching with the complete assembled sequences. The second column shows you the length of these assembled sequences, and you can see roughly these are 5 million base pair long. And this is the statistic of the short read data that we are using. So these are the coverages of the short read datasets that are available publicly. As a pangenome graph, we are using a graph constructed from 49 complete MHC sequences that are available from HPRC consortium. And these 5 haplotypes that are part of the test framework are not part of the pangenome graph. So because that would imply that the problem would be much easier. So these 5 haplotypes are not part of the pangenome graph. Okay, so I hope this experimental setup is clear now. Let me show you how this method performs in terms of the concordance with the ground truth. And to measure the genotyping accuracy, what I'm going to do is I'm going to check the sequence path that I inferred from the pangenome graph. What is its eddy distance from the ground truth MHC sequence? So that is, I'm comparing 2 sequences of approximately 5 million bases long, and I'm checking their eddy distance. Lower is better because eddy distance of 0 means that they're exactly identical, and a higher eddy distance means that there is some discordance between the ground truth and the reconstructed haplotype. So we are using 3 methods: phi, VG, and Pangenome. So in Pangenome, for example, it produces a VCF file in the end, and we convert that VCF file by incorporating those variants in the reference haplotype so that we get the inferred haplotype as well. So I'm showing you these results for 5 haplotypes that we were using as part of the test framework. And I'm also using different coverage values. That is, what happens with 0.1x coverage, what happens with 0.5x coverage data, up to the complete data. So because we want to evaluate how well this genotyping framework is working at different coverage levels. So these are the results that we find for all these 3 methods. And the key observation is that the proposed implementation is comparable to the existing solutions at high coverage. So for example, here, when we compare against VG personalized pangenome graph construction, then in this case, the results were almost comparable with VG at high coverage values. So you can see a similar values, and remember that the y-axis are in log range. So even small differences in y-axis will be large gaps in the absolute scale. So at high coverage, these methods are roughly comparable, but at low coverage is where we see our method to be competing better with other methods. So phi results in better reconstruction of the haplotype, at 0.1x coverage, 0.5x coverage, and even at 1x coverage across all 5 haplotypes. And this may be because we are not we are directly using this k-mer absence presence in our formulation. We are not using any heuristics, and we are not using k-mer counts in our formulation. We are directly using the k-mer presence or absence, because if you think of from low coverage data, other methods are using k-mer counts to predict whether a k-mer is homozygous, heterozygous, and so on, which can be misleading. So, for example, determining whether a k-mer is an error k-mer or whether a k-mer is a homozygous or a heterozygous k-mer, that can be difficult with low coverage data. And as a result, that's where we see a significant advantage compared to other methods in haplotype reconstruction. So moving on, we also check, okay, what is the impact when we keep on increasing the Pangenome graph size? That is, what if you if you use a smaller Pangenome graph with only 3 haplotypes, all the way to the Pangenome graph with 49 haplotypes? What we find is that adding more Pangenome graphs helps with the reconstruction accuracy. So you can see that the edit distance keeps on improving as you add more number of haplotypes. However, at the same time, we also find that the runtime and memory usage of our methods are increasing with the increasing number of haplotypes. And this is something that we would like to focus on, is how do we scale this solution to large number of haplotypes going forward. As I mentioned earlier, that we also optimize the integer programming framework by relaxing the edge variables. And here I'm showing you that relaxation helps. For example, in the quadratic programming framework, we get 1.6x speed up. And in linear programming framework, we get 1.12x speed up using this variable relaxation. So all these optimizations that we put in the integer programming ultimately helped us scale to these MHC sequences because we are working with millions of variables and a large number of constraints in this programming framework. So to summarize, I showed you a novel problem formulation for haplotype inference from pangenome graphs, which can be useful for genotyping applications. And this can address genotyping all kinds of variants, for example, SNPs, indels, large variants, and so on. We show that the path inference problem is NP hard through reduction from Hamiltonian path problem. And subsequently, we developed efficient solutions using integer programming that can scale to the regions of interest in human genome. For example, through the experimentally I showed you that our algorithm can reconstruct MHC sequences with low edit distance from the ground truth sequence. So for example, using only 0.1x coverage, the reconstructed sequence are up to 99.96% identical to the ground truth sequence. And you are welcome to use file. It's freely available on GitHub. As a future work, one obvious thing that we have to do here is to extend this framework to deployed samples as well. And the idea there would be to somehow extend this problem from inferring one flow, like having a flow, ST flow of one. So right now we have a ST flow of one that we are looking at. In the deployed case, we would want a flow of two from source to sink so that we get two paths from the Pangenome graph. We also would like to improve scalability to larger graph and larger number of haplotypes. And finally, we are exploring the extension to metagenomics and clinical genomics, where genotyping using low coverage inputs would be useful. With that, let me also acknowledge all the co-authors of this work. So this work was led by Ghanshyam Chandra, a student in my group. And this is an excellent collaboration with Daniel Gibney and his lab, as well as Alexander Dizzy and his student, Stephen. So with that, I thank you, and I'm happy to take any questions.
So thank you very much. It was a very nice and very clear talk. I will start with the first question, but I'm inviting everyone else to ask question on the metrics channel. The question is, so you said that the problem is NP hard when the cost is the sum of the number of recombinations and the number of chimers that you're not able to find.
Yes.
Uh, I expect that it is still NPR even if you consider only the recombinations or only the chimers that are missing.
So if suppose I only consider recombinations, then basically I would output any one of the haplotypes, right? So I would output you, let's say, if there are three haplotypes in the Pangenome graph, I would directly output one of the three haplotypes and say their recombinations are zero. Understand what I mean? So I can easily output a path where recombinations are zero.
Mm. Oh, I see, I see, I see, I see, I see. Okay, okay. Thank you very much. And okay, let's see if there is any question. Any more question? And the second question that I have. So you mentioned at the end that you plan to extend it to diploid organisms. What is the added difficulty with respect to the haploid organisms that you have?
So there are two like there's so one key difficulty there is that when we are predicting two paths, uh, my understanding is that that the ILP becomes much more complex. That is, it has to do more work. The optimization space, the search space is increasing now because we now instead of searching one path, it has to search for two paths. So, so ILP would take much longer to solve. That is one. But then also from a practical point of view, we would have to make sure that like even if we are producing two haplotype paths to the giving two haplotype paths to the end user, we would have to see how many switch errors are happening. Like because when you move to deployed genome, when we are giving two haplotype paths, that is we are giving the genotypes as well as the phasing information, and therefore we have to there not only the genotyping accuracy but also the phasing accuracy would matter. Does that make sense? So because when I
Okay.
give two paths, it may happen that the two paths are like switching between each other wherever there is like whenever let's say there is no heterozygous information or there is not enough information to distinguish between the two paths, then it might happen that I can switch between the
Yeah,
haplotypes.
that is very clear. So I see a question in the chat. Thank you for the very nice talk. I missed something in the expanded genome graph concept when you add intermediate vertices to represent recombinations. Could you please explain again the idea and why it enables to scale? I understand somewhere there is the idea to linearize, but I don't get the point.
So, so if you remember there, if we don't add those intermediate vertices, then we have to introduce all to all edges in the worst case. So in the worst case, there may be scenarios where there may be all to all connections among the haplotypes, and that's where the number of connections can be quadratic in number of the haplotypes. But when you put a intermediate vertex, then what can happen now? What can I do is all the source vertices which want to recombine, I will connect them to that intermediate vertex, and then I will connect the intermediate vertex to the target haplotypes. So then what happens now is that in the worst case, the number of connections is linear in terms of the number of haplotypes because now all the haplotypes go to the first the intermediate vertices, and the intermediate vertex then connects to the following haplotypes. So then it's like a all to one and then one to all in the worst case.
Okay. Now since this talk and this discussion is streamed on YouTube, but there is a bit of delay, so we have to wait a
Okay.
few seconds to understand. But everything was clear. But I see someone else is preparing another question. So
So I forgot to say that this paper is up on bioarchive as well. So if people want to read it further, they are welcome to search for the title on online, and there will be a preprint on this.
That's perfect, and that's perfect. Let's see the question that is coming, because I see two two people are typing, and then I need them to send the complete question, and then I'm going to. So how this approach would fare with error-prone reads and sometimes shorter than conventional aDNA datasets? What would be the constraints?
So the first question part, I understood that if there are errors in the short reads, because we are using publicly available short reads, which also have some level of errors in them. Uh, but the errors will not hurt our, like erroneous schemas will not hurt our formulation, because what will end up happening is that most of those error schemas having errors will not match to the graph at all. So they are not part of the objective formulation. So the optimization problem formulation will basically ignore those schemas, because there's no way we can have the inferred path going including those schemas in the uh, in the graph. So because if the so this is assuming that the k-mer with error has no match in the graph. However, in rare instances, if the k-mer with an error has a match in the graph, that's when the errors would hurt. But in practice, what we find is that most of the k-mers with errors don't have a match in the graph.
Okay,
The
okay.
second part is this, where we this the
It again, so you mentioned aDNA datasets, and what would be the constraint? So if you need to if add more constraints to the ILP formulations, or if you don't expect to be able to manage in practice error rates that are too large, but you have answered that. You have answered that before. So let's see if there is another question.
the errors would also make we would need to keep our k-mer length reasonably, like we cannot extend the k-mer length too much, because then there's a good chance that every k-mer would like every k-mer would be having error. So that's, yeah, that's one other trade-off that errors would introduce. That if the errors are too many, then we would need to maybe keep the k-mer size short.
So one thing that maybe I missed in your presentation, but how long does it take to actually run Groby on the largest instances?
So let me see. I think it takes 2 to 3 hours with all the optimizations.
So several of hours, not days.
Yes, yes, yeah. So
Which is very good.
because we have two frameworks. One is quadratic constraints and one is linear constraints.
Yep.
What we see is that the quadratic constraints runs faster. Groby solves the optimization problem when we have quadratic constraints much faster, but there the memory usage is higher. So it's a runtime memory trade-off that which we see from Groby.
Okay, so I don't see any more questions. So thanks again. It was a very, very nice talk. So
Thank
thanks
you.
again, and we hope to meet everyone in person one of these times.
Thank you, Simon. Thank you
Thanks
very much.
again. Thanks
Thank you
again.
very much.
Next talk will be in 10 minutes. Hello.
Hello.
Hello. Hi. The volume is very low, but
Does that help?
yes,
Yeah,
yes,
a
a lot.
lot.
Much better. Nice to meet you, and thanks for having accepted our invitation. First of
Yeah,
all.
thank you for the invitation.
So, my suggestion is that you try to present, and so we see if everything works.
Is that working?
Okay, I think. Okay, yes. May you try to start the presentation mode and switch to a different slide so that we check if everything works.
Okay.
Okay, please change slide. Okay, okay, everything works. So that's perfect.
Okay. Cool.
Okay. So I know it's quite early there. Okay.
Oh, I'm actually
Uh,
living in France now, so it's
oh, I
8:10.
see. I see. So, but are you still a PhD student at UC Santa Cruz?
Yeah,
Okay,
I'm just
okay. I just
finishing up remotely.
okay, 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 okay. Oh, that's good. That's good. No, I just wanted to be sure that. So, how do you pronounce your name?
Shen?
Shen?
Yeah,
Shen, and the surname?
Chang.
Shen Cheng. Okay,
Yeah.
I will try to do my best. I apologize in advance for the errors
Yeah,
that
no
I'm
problem.
certainly going to make. Um, okay, okay. So we are all set up, and we have 7 minutes. So, so that's fine. Uh, I mean, the next speaker is going to be in an hour, so if it takes you for even 45 minutes to give the presentation, that's fine, and there's plenty of time, so
Okay.
don't worry about anything. And we are going to collect the questions via the Matrix chat. The talk is streamed on YouTube, and that means that there is roughly 1 minute delay from when you present and when the audience will be able to see your presentation. So after your talk, we probably I will invite questions even during the talk. But what we have seen so far is that there is essentially a minute gap between the end of the talk and when the questions arrive. So just to let you know that there is nothing wrong there, but that's just the technological thing. And so how long have you been in France?
Um, about a year and a half.
Oh, I see.
I was expecting to finish my PhD in a couple months after I moved here, but it's taken a lot longer than that.
And they've already found your next
No,
position.
not yet. I have to start looking, but um, I want to find something in France, so it's
I see.
a little more
Well,
limited than I'd be in the US, especially in California.
yes, and when you start focusing on a specific place, it's usually more competitive to find something, but I'm sure you'll be able to find
Yeah,
some good offers. Sure. But I understand that it's can be it can be stressful, especially if you're now preparing your PhD defense, and that's also a busy time of your life.
yeah.
So I
I
wish
would
you well.
prefer to just thank you. I would prefer to just stay a PhD student. It's a great job. Don't have a lot of responsibilities. I can just work on.
Yes, but at certain point you also start getting fed up with the salary of a PhD student, which is not as
Yeah, that is true.
attractive. So, okay, so 3 minutes. One minute. Um, Okay. If we are ready, we can go.
Okay.
Okay. So I'm very glad to have Chen Cheng here to give a talk on the long read mapping against the Pangenome graph. Chen is a PhD student at the University of California, Santa Cruz, and I'm very eager to watch her talk here. Thanks again for being here, and please go on with your talk.
Okay, yeah, thanks for the invitation to come speak today. I'm Shen. I'm a grad student in Benedict Payton's lab at UCSC. And I'm part of the VG team. So VG is a big unwieldy toolkit of lots of different pangenome tools. So we can do things like construct a graph, manipulate a graph, find subgraphs, get graph statistics. And we can also do some kinds of analysis. So we can map reads. We've got 3 different mappers. And then we can also do downstream analyses like genotyping variants and doing RNA analysis. But my primary project has been VG Giraffe, which is one of our 3 mappers in VG. This is the most recent one, the most, I think, multi-purpose mapper. We published this a couple years ago, and we showed that it was very fast, pretty accurate, and was pretty good with variant calling. But it was really designed for short reads and wasn't great with longer reads. So the algorithm for the original short read giraffe was very targeted to short, highly accurate reads. And when we applied it to longer reads, which are very long and very error prone, it wasn't great. It was super slow, used a lot of memory, and kind of killed our computers. So there are existing long read mappers that do work. So I think GraphAligner and MiniGraph are probably the most well-known. There's also GraphChainer and Paneliner, which use components of GraphAligner and MiniGraph respectively. But each of these also has their own limitations. So GraphAligner tends to be really slow, particularly as the graph gets more complicated. And MiniGraph was designed to really only align reads to graphs constructed with MiniGraph. So MiniGraph graphs don't include smaller variants. So it's limited in that it can only really align to very simple graphs. So we wanted to make a new long read mapper that would be both fast and generalizable to more complicated graphs. And since we already had a short read mapper, we started with adapting that. So in VG, we work in the variation graph model. So we have a pangenome, which is basically just a collection of haplotypes. And from those haplotypes, we use a graph construction method to get a variation graph out of it. And the variation graph model incorporates both the graph itself, so these nodes and edges, as well as the haplotypes. So the original haplotypes in the pangenome can be represented as paths through the graph. And these are the file formats that you might see if you use VG. So anything ending in G basically is a representation of the variation graph itself. And then the GBWT, which stands for Graph Burrows Wheeler Transform, represents the full set of haplotypes. And I threw in a bunch of citations throughout the presentation if anyone's interested in background reading. Okay, so this is the input to our giraffe algorithm. So we've got a read up here, which I'm representing as a bunch of colored blocks that correspond to where they would align in the variation graph. So the original giraffe algorithm follows a pretty standard mapping framework that pretty much all mappers use. So seed, cluster, extend, everyone uses it pretty much. So the first step of seeding, we want to find very short alignments between the read and our reference. So we call these seed alignments. They're just short matches. Um, and for the original giraffe algorithm, we used a minimizer index, which is basically a dictionary mapping a k-mer sequence from onto the haplotypes of the reference pangenome. So pretty simple. We didn't change this in the long read algorithm. The next step is clustering. So we take all of those seeds that are scattered throughout the reference and we try to partition them into groups of seeds that are close to each other on the reference pangenome. And since distance is kind of a hard problem in a graph context, we use this distance index to figure out how far away the seeds are from each other. And this is a useful step because it helps us focus in on which regions of the genome the read is more likely to map to. So if you've got a big cluster with lots of seeds, it's more likely that the read goes there than somewhere else where there's fewer seeds. And then in the original giraffe algorithm, we take those seeds and we go to the haplotypes to do alignment. So within a single seed in the cluster, we extend the alignment as far as we can along the haplotypes without allowing any gaps. And this is the step that really made the original giraffe algorithm very fast for short reads, because we expect that the reads are short and pretty much error-free, or at least they don't have a lot of indel errors. And we also expect the haplotypes to cover most common variants that we find in the human population. So because of this, we would expect the read to match exactly at least one of the haplotypes in the graph, meaning that we can find the entire length of the read at this gapless extension step. If we don't find it at gapless extension, we'd go on to continue the alignment in graph space, which is a much slower and memory-intensive process. And so this is why the short read algorithm didn't really work for long reads, because as we see here, it's less likely that the entire read is going to match exactly one haplotype. So we do a lot less gapless extension relative to the length of the read. And we spend almost all of our time trying to align the read to the graph itself, which really exploded the memory use of the algorithm. So for our long read algorithm, we went back and changed our clustering step, where we just find a big mass of seeds, to a chaining step. So chaining is also a pretty standard algorithm for long read mapping. The goal is to find an ordered set of seeds that are collinear in the read and the reference. So that means that they occur in the same order in the read and the reference. So in these little alignment blocks, none of them overlap. You can walk from one to the other in the same order in both the read and the reference. And another thing we found was that the distance index that we used to calculate minimum distance wasn't fast enough for chaining. So we introduced a new data structure called a zip code tree that I'll talk about more later. So now that we've got a chain of seeds, this forms the backbone of an alignment. And now we have to do alignment. So pretty standard. We take the seeds from the chain, project them onto the haplotypes. And then we do alignment in between the seeds and out from the ends of the seeds. And we try to make this as haplotype consistent as possible. So again, we start trying to do alignment along the haplotypes until we hit a point where they're no longer haplotype consistent. So here in this example, these two alignments can't reach each other on any of the haplotypes. So we go back to graph space and do alignment there. Yeah, and doing that, we can get the full alignment. So this is an outline of the final long-range giraffe algorithm, pretty standard. And then the data structures that we need to support all of the queries we make. And if you ever actually run giraffe, you might notice that there's a lot of indexes that we require. We get a lot of complaints about that, but this is why we need lots of indexes to make it fast. And these are all the different file formats that we require. So the GBZ contains both the haplotypes and the graph, but you also need the minimizers, the distance index, and now this new zip code thing. Right, so for this presentation, I'm going to be focusing on the chaining step because it's the part that I worked on, and I think it's also the most novel of all of these steps. Right, so chaining, again, is a pretty standard problem. We're given a set of seed alignments between a read and a reference, and we want to narrow that down to an ordered set that are collinear in the read and the reference, so none of these overlaps. In a linear context, this is usually found using dynamic programming. So this is an algorithmic technique to kind of break down a problem into subproblems. For chaining, the idea is you want to find the optimal chaining score of a chain that ends at a particular seed. So if we order these, we want to find the optimal chaining score for this seed. But as we go through the algorithm, we assume that we've already found the optimal chaining score for all previous seeds. So we found the chaining score for the best chain that ends at this seed, the seed, the seed, the seed. Right. So this is the dynamic programming recursion for Minimap2, which I've cited up here. They're generally pretty similar, so I just used this one as a common example. So what this formula means is the chaining score for any seed I, as we step through the algorithm, is going to be the maximum of this formula for all previous seeds j. So we check each one of these individually. And the actual formula for the score here, this is the chaining score for seed j plus the number of matches between I and j. So we look at the sequence between the seeds in the read and in the reference and count up the number of matches. And we subtract from that the gap cost between I and j. So the same thing, but we look for the gaps. The gap cost definition can vary between different algorithms, but in general, both the gap cost and the matches depend on the distance between the two seeds in the read and the reference. So in a linear context, that's pretty easy to calculate because we know the offset of each of the seeds in the read and in the graph, in the reference. But in a graph context, that becomes a much more difficult problem because distance and reachability are both not very well defined in a graph. So as an example, between this orange seed and this purple seed, they're not reachable in the graph, so we can't order them and we can't really find a distance. And more problematically, we often see cycles in a graph, in a variation graph. And those are really not easy to deal with in a dynamic programming context because when we want to find the distance from one of these green seeds to the other, the distance isn't well defined because of this loop here. We can take this loop an arbitrary number of times and just keep increasing the distance between the two seeds. So there has been some previous work done on chaining in a graph. So these two, GraphChainer and Paneliner, are two mappers that have used their own chaining algorithm. One of them is from Chirag Jain. He's got a whole series of papers that are really great on chaining. If you read them, I would recommend reading them in order because I didn't at first and it was quite difficult to follow. But I think both of these chaining algorithms are based on this original one for sparse dynamic programming in DAGs, DAGs being directed acyclic graphs, so it doesn't include these cycles here. But the basic idea here is that we take a path cover of the graph, so we find like linear paths and then use those linear paths to predict or to estimate distances in the graph. So if you're interested in this subject, this is a great place to start. But for giraffe, we already have an idea of minimum distance that we've been using. So we used that as a starting point for our training algorithm. And to understand the distances that we used, I'm going to have to go into a little bit of background. So to start out, I'm going to introduce the snarl decomposition. So snarls are a method of finding organization in graphs, because graphs can get really big and complicated. Snarls help us simplify them and think about them in more distinct pieces. So formally, a snarl is a subgraph of the graph that's disconnected from the graph or connected to the graph by two boundary nodes. More conceptually, you can think of a snarl as a site of variation. So in this purple snarl here, you can take either of these paths through the snarl, representing two different variants in your original genome. And then the two boundary nodes represent common sequences. So this here is a snarl representing two different variants. Oops. This one here is a snarl representing a duplication. So as you walk through this graph, you can take the duplication edge and walk through the node multiple times. And snarls generally occur consecutively along the pangenome, and we call a sequence of nodes and snarls that share a boundary node a chain. And we describe the organization of snarls and chains in the graph using a snarl tree or snarl decomposition. So here, this gray chain containing everything has two child snarls, this purple one here and this green one here. And then snarls can also be more deeply nested than that. So for example, in this snarl, it has two children. So there's kind of two paths you can take, right? There's this node here, and then you can take this path, which in turn contains another snarl. So this is how we break down the graph into smaller and smaller pieces that we can describe using a snarl tree. And for completeness, this is what the full snarl tree would look like. Each node in the graph is the child of a chain, and sometimes the only child of a chain. So this purple node is its own chain. But for the sake of this presentation and keeping things on a single slide, I'm just going to represent it like this. So the other useful property of the snarl decomposition is that we can kind of eliminate some of the complexity of the graph by picturing a snarl as only containing its child chains. So from the perspective of this purple snarl here, even though there's all this nested variation down here, we can imagine that it only contains two very simple children, and then it becomes this simple bubble. And because of this property that snarls are separable, they're only connected through their two boundary nodes. Doing this doesn't change the topology of the graph, right? It's still this pretty simple bubble that it would be like this. Right, so that's this property here. Snarls and chains are only reachable through their boundary nodes, and this property is useful for calculating distances because when we look for the distance, the minimum distance at least, between two positions on the graph, the minimum distance from anything within a snarl can be broken to anything outside of the snarl can be broken up into the minimum distance from within the snarl to the boundary of the snarl and the minimum distance from the boundary of the snarl to the second position. And that's because there's only 2 points through which you can exit the snarl. So technically you could also go this way, but there isn't a path there. Okay, so that property is the basis of our snarl distance index. So this is what we used in the original short-read giraffe algorithm for calculating distances. The idea is that we break up a minimum distance calculation along the snarl tree. So to go from here to here, we use an index to find the distance from every point along every snarl and chain to the boundary of its parent. So from the position to the boundary of the snarl, from the boundary of the snarl to the boundary of the chain, and so on, walking up the snarl tree for each position, and then we find the distance between the children of the common ancestor. And in this example, because I'm limited to the size of a slide, it doesn't look like this is a big win. But if you look at an actual graph, this snarl-based distance metric is its complexity is linear in the depth of the snarl tree, which tends to be pretty shallow, versus something like Dijkstra's, where the complexity is based on the size of the graph. So this is much faster. In practice, it's constant time. But when we used it for chaining, we found that it wasn't fast enough. This is really a problem with computers, because the problem is really all the memory accesses, because we stored a huge index for each snarl and chain, and we had to go look up stuff in that big index every time. So we've created this new, smaller data structure called a zip code for calculating distances between positions. So we store one zip code for each node, and the zip code contains a minimal amount of information to be able to calculate distances between two zip codes. So it stores the path to reach it in the snarl tree, so chain, snarl, where it is in the snarl tree. And then for each snarl and chain, it contains the distance index, the distance information you'd need to calculate distances. So like the distance from the snarl to the end of the chain, the distance from the chain to the boundaries of its parent snarl, etc. So finally, getting back to chaining, we use those zip codes to create this zip code tree, and the goal is to be able to quickly calculate distances between seeds in a graph, with the goal of chaining. And ideally, since we want to make giraffe generalizable, we want to be able to handle duplications and inversions and all that topological complexity that we see in graphs. So the intuition behind zip code trees is that we want to make a subgraph of the original graph with the seed positions, such that we can calculate distances between the seeds in a traversal of the subgraph. And in order for it to be used with chaining, with the dynamic programming algorithm, it should be a directed acyclic graph, or a DAG, which means that there's no cycles, none of that weird complexity that we tend to see in real variation graphs. So to create this zip code tree structure, the input is seed positions on a graph, and each of the seeds has a zip code associated with it, so we can quickly find distances. And we're going to use the snarl tree kind of as a guide for creating the zip code tree. So first, we make one node in the zip code tree for each seed in the graph. So pretty simple. Each seed here is a node in the zip code tree. And then we add a node for the start and end bound of every snarl and chain that contains a seed. So this chain is the big root chain containing everything. We add its start and end bound. Then we do that for the child snarls here. So the start of the snarl, the end of the snarl here and here. And we do that walking down the snarl tree for everything containing a seed. And technically there'd be another chain here, but I ran out of space. Okay, the next step is to order the snarls, the children of snarls. So in a graph, these children of the snarl are unordered, but we assign it an order in the zip code tree. And when we can, this is a topological ordering of the snarl children when the snarls are DAGs. When they're not, we just kind of guess at a topological ordering. And then once we have that ordering of the children, we can add in edges. So the edges occur between nodes that are adjacent in the graph or in the snarl tree. So between children of the chain, we add in an edge. And then between children of the snarl, we add an edge. And for snarls, since we've ordered the children in a topological ordering, we add an edge from each child to every child that came before it in the topological ordering. So again, for a DAG, that would capture the full connectivity of the snarl because it's in a topological ordering. But if the snarl isn't a DAG, so in this case, the snarl isn't acyclic. You've got this looping edge here, and just adding edges like this won't capture that looping edge. And we want, as I said before, we want these zip code trees to be acyclic because it makes everything easier. But we also wanted to capture the topology of this graph. So to sort of include that duplication edge, we include information from the read. So we allow these nodes that break the ordering of the read to split up into two separate children. So even though in the original graph, these two seeds came from a single node, in the zip code tree, we allow them to split up into separate children of the snarl. And then we order those separate children based on the ordering in the read. So this one comes first, and then we add edges as we would for a normal snarl. So this is the final structure of the zip code tree. For the sake of the presentation, again, I'm simplifying it. The topology is pretty much the same. I just took out the some of the chain boundaries. And then in the actual data structure, we store the zip code tree as this one linear vector of nodes and edges. And this improves our memory colocality. So, when you're looking things up in memory, it's faster to look things up that are close together in memory versus far away. And so when we go to find distances in a traversal of the graph, everything we're looking for is pretty close together in this linear representation. So yeah, we find distances in a traversal of the graph, and we add up the distances on the edges to find the distance between seeds. And in doing this, we can use these distances for the purpose of chaining. So now if we've got these seed alignments between the read and the graph, or this zip code tree, we can find the best chain of seeds by walking through the zip code tree and finding the graph distances from the zip code tree. And this would be the ordering of seeds from the distances in the zip code tree. And the nice thing about the zip code trees is that even though they're acyclic, they're pretty simple. There's no loops or inversions or anything. We can find this path here, which in the original graph corresponds to this looping path here, which we wouldn't have been able to find using the original graph because we can't really deal with loops like that. So that's how zip code trees deal with duplications. We can also deal with inversions, again, by taking information from the read. So in this graph, this motif here represents an inversion. You can traverse this node going forwards, or you can take these inversion edges and traverse it going backwards. So using the information from the read about orientation, we can tell that we need to traverse this seed going that way. And then when we go to make the zip code tree, we can make sure that we include the seed in the correct orientation. Right, so that's how we do chaining. We use the zip code tree for distances. And because the zip code tree is created for a set of seeds, we do zip code tree construction once per read. So that adds a new step to the algorithm. So now it's seeding, create the zip code tree for calculating distances, And then we use the zip code tree for chaining, and then we do alignment. And alignment isn't really a new topic, so I'm not really going to go into it. But if you're interested, these are the graph aligners that we use in Giraffe. So we've very recently merged our long-range Giraffe algorithm into the main branch of our GitHub repo, meaning that it'll be available in the next release of VG, which should be soon, I think. And running it is pretty easy. We keep the same interface, but we now have a new option for running long reads. We just use a different parameter preset. And then we've also added this new index, the zip codes. So that can be built with VG auto index like normal. And so here are some preliminary results. This is showing the runtime and memory use of different long-read aligners on hi-fi and nanopore reads. There's a bunch of different graphs and aligners, so I think we should focus in on this one here. It's the most comparable. This is it's kind of poorly named. The HPRC graph is actually this whole mess here. So what it's saying is it's from the Human Pangenome Reference Consortium version 1.1 of all of the data constructed with mini-graph cactus using CHM13 as the base reference genome. And the D9 means that we've frequency filtered the variants. So out of the 90 or so haplotypes, any variant has to be in at least 9 of them to be included in the graph or something like that. So it just takes out the very uncommon variants to take out a lot of the complexity. And for short-read Giraffe, if you're not doing haplotype sampling, this is the graph we recommend using. So yeah, here I'm comparing Giraffe and Graph Aligner, mapping to to that graph. And the conclusion is that Giraffe, is much, much faster than Graph Aligner. So it's actually faster than this graph would make it seem, because we had problems running with Graph Aligner. It took more than like 5 days, which is the limit for running things on our server. So it got kicked off, and I had to rerun it with twice the number of threads. So to make this a fair comparison, I think the Graph Aligner numbers should be doubled or so. But in any case, Giraffe is much faster than Graph Aligner. We do use quite a bit of memory, and again, to be fair, the memory used for Graph Aligner should probably be halved. But yeah, we're still comparable to the other mappers. In terms of accuracy, we're pretty accurate here. I'm showing rock plots on 1 million simulated reads. And again, there's lots of different conditions here. So I'll just highlight Giraffe and GraphLiner against the same graph, and then Mini-Map2 mapped against CHM13. And we're more accurate in terms of just the number of reads mapped correctly. And we also have better mapping quality calibration. So mapping quality is supposed to be a measurement of how confident we are that the read got mapped to the correct place. Mapping quality of 60 means that we expect there to be a 1 in a million chance that the read was incorrectly mapped. So for 1 million simulated reads, we would expect to get 1 incorrect map U60. We actually get 150 and 1,500, which is not great, but it's still better than GraphLiner and Minimap2. So good accuracy, better mapping quality calibration than the competition, but still not great. And simulated read accuracy is not like the most robust measurement of actual performance. So we also did variant calling. So here I'm showing small variant calling with deep variant, and then structural variant genotyping with VG call. And then for the small variants, I'm comparing against Minimap2 in orange. And then for structural variants, I'm comparing to Graph Aligner in purple. And here I'm just showing the F1 value as like an overall measurement of how we're doing with variant calling. And in general, the blue points representing giraffe are doing better than the other points, except for indel calling on hi-fi reads. Minimap 2 is doing better than us, but we're not too worried about this because for short reads, when we ran deep variant, we trained a model to specifically use giraffe reads, and we saw a really big improvement with the trained model. But for long reads, we haven't done that yet. This is just with the default model, which is trained on Minimap 2. So we expect that when we train a model, we'll improve. So not too worried about this gap. And yeah, that's all I have. So giraffe, we think, is in a place where we're ready to share it with the public. It'll be in the next VG release, but we haven't published yet. We're still waiting on some new results. There's going to be a new set of HPRC graphs released soon, so we want to test giraffe on those graphs to make sure that it's robust to the bigger and more complicated graphs with more genomes in them. For short reads, we now recommend haplotype sampling as the recommended pipeline. But it doesn't work as well as we expect it to with long reads, so we're still debugging that a bit. And the long read code path with chaining doesn't work on paired end reads yet. We still use the old code path for that. So just for the sake of keeping our code clean, we're planning on extending long read giraffe to work with paired end short reads. And then we need to publish, and I need to graduate and get a job. So that's it. Thanks to my whole lab, everyone in this picture, and particularly the people highlighted who are working on this project with me. And thanks for listening, and I'll take any questions.
Thank you very much for your talk. It was very nice and very clear. We already have some questions from the audience. So I'm reading the first question. Thanks for the very interesting talk. To better understand the results, do you know more or less the dimensions of the graph, number of nodes, number of beings, and so on? Apologize if I missed those data.
Oh, number of nodes and edges, I don't know. It's I think 92 haplotypes though. Yeah, I don't have a good sense of the node count.
Okay. And another question is, what is the definition of correct alignment in the preliminary results?
Yeah, that's a good question. So when we simulate reads, we label the reads with where they come from we simulate from a graph containing the sample we want to simulate from, and then also the reference. And then using that graph, we can project the reference coordinates from the graph onto the simulated reads based on the graph alignment. So all of our reads have graph coordinates, or reference coordinates of where we expect them to map in the reference genome. And then we can do the same thing when we map them to our actual pangenome. We can project the reads we can project the reference coordinates onto the reads to see where they overlap with the reference. And then we use those reference coordinates to calculate accuracy. So anytime the read has any reference coordinate that's correct anywhere in the read, we count it as correct. So it's a little bit permissive in that anytime the read overlaps anywhere with the correct placement in the reference, it's counted as correct.
Okay. And how do you simulate the reads?
Yeah, so VG has its own read simulator. I don't know how exactly it works, but we create a graph containing the reference genome and then the sample we want, and then we simulate from the sample.
And do you have any idea of the maximum error rate that you are able to manage?
Oh, that's a good question. I'm not sure. I think we've tested it on D9 or sorry, R9 reads. and it worked. It wasn't great, but yeah, we haven't tested it on ancient DNA or something with a lot of errors. But I imagine that as we add more and more errors, it's just gonna use more memory. So it depends a lot on how much memory you have available.
So you think it's more a matter of the memory that you need?
Yeah.
I see. I see. Thank you. Let's see if there is any more question from the audience. Wait a little bit, but okay. So it does not look like there is any more questions. So thanks again. I really liked your talk. It was very, very nice, very clear, and so I wish you well for your job hunt.
Thank you.
Okay, thanks again. So the next talk will be in 20 minutes. Davide, benvenuto.
Hey, can you hear me?
Yes, perfectly.
I think now you can also see me.
Yes, and now we can also see you.
Yeah.
We can test if everything works. So
Yeah.
if you can prepare to share
Yeah.
screen presentation.
I can. Yeah. Yeah, you should be able to like,
Okay, now we see your screen. Yes, that's the first slide. And move to the next slide. Okay, that's
yeah, you
perfect.
should be able to.
Okay,
Good.
we are all set up and we have 18 minutes. So just okay. Is it correct that you're now senior bioinformatician at Human Technopole? That's so when I want to introduce you.
Sorry, just a second. I'm like closing the YouTube stream I had on that because otherwise it overlaps and it's like a bit of a mess. Yeah, I need to go back to this streaming here. Okay. They're a bit lagging to me, but probably that's a problem with my connection.
Okay, so the stream on YouTube is about 1 minute late.
No, I mean, so your voice is lagging to me, so I don't know. It's probably a problem with my connection. Well, I don't know. Let me check what happens.
In any case, that's not a problem. I mean, we are listening to you perfectly. The presentation is fine. So I only need to introduce you. And so is it fine if I say that you have got your PhD from University of Siena and you have worked in several institutions in Italy and Europe, and you're now senior bioinformatician at Human Technopole?
Yeah, it's perfectly fine.
Okay. Okay. So we have 15 minutes. If we want to take a break, there's plenty of time and everything is set up. as you wish. Okay. We have 2 minutes. So if you are ready, we can start. Okay, okay, perfect. So it's my pleasure to have Davide Bolognini here today. Davide has got his PhD from the University of Siena, and afterwards he has worked in several institutions in Italy and in Europe. Now he is a senior bioinformatician at the Human Technopole, so it's close to us in Milano, and is going to talk about the inference of complex haplotypes from sequencing in a pangenomic setting. So, Davide, thanks again for having accepted our invitation, and please go with it.
Yeah, hope you can hear me well. So, yeah, good morning, everyone. And first off, I'd like to thank the organizers for having me here. So, my name is Davide, as Gianluca was mentioning. I'm a senior member of the Genome Analysis Unit, which is a unit within the Population and Medical Genomics Research Center at the Human Technopole in Milan. We offer computational support to groups both within and across centers at the Technopole. And beside the computational support activity, we are also involved in several research projects. So more specifically, I'm the one dedicated to projects involving long read sequencing and pangenome related stuff, and that's why I'm here. Today, I'm gonna talk about the topic that you see here in the title. Sorry, so I have a different screen for the presentation. That is approaches in general that exploit the pangenome idea to improve on genotyping. So the first part is a bit didactical, so be with me for that. I've no idea of how the others tackled the presentations problem, but yeah, so I will be very quick on the first part, and then I'll go on with a couple of approaches that I think are interesting. So, in general, what is genotyping? Diploid organisms have two copies of each autosomal chromosome, each of which can carry genetic variation. So, the process of determining whether a certain variant allele is located on none, one, or both of these copies is called genotyping. So, for instance, here you have the schema of a sample with two haplotypes. Yeah? And I have highlighted three alleles, the green one, which is present in both haplotypes, the yellow and the blue ones, which are present just in one. So routinely used genotyping methods for sequencing data are based, for instance, on short-read alignments to the reference genome. And I have highlighted here the alleles that the reference genome exhibits. So you have the red allele, I hope you can see my pointer, and the blue allele here. Yeah. So and this what you see here is the possible alignment of the sample reads to the reference. Yeah. So in the first block there is no variation highlighted. So you would say that in that region the sample haplotypes are both equal to the reference. So we can call RR. In the second block the reference is red, but our sample only has green reads, so we infer that both the haplotypes are alternate, so AA. Again, a third block which is equal to the reference, and then the fourth block is blue in the reference, but there is a mix of yellow and blue in the sample, so we derive that one haplotype is reference and the other one is alternate. And again, a final block which is equal to the reference. So again, reference. So this is usually represented in the VCF specification with the following entries. Yeah. So you have 0, 0, 1, 1 for alternate, and so on. So this is the routine way to do genotyping from short reads. And this is the best case scenario where you still are able to somehow identify the genotype of an individual, even if not all the alleles are present in the reference. Yeah? These genotyping methods, however, suffer from the so-called reference bias, so which occurs when reads originating from non-reference alleles fail to align to their true point of origin missing in the reference, or those reads align to a different locus. So for instance, here the yellow reads fail to align and they are left unmapped here, like on the right side of this slide, because the yellow region is not represented in the reference genome, and this causes wrong genotyping results in effect. So you may infer that here the sample is reference, but in effect, he is not. So one possibility would indeed be to use many references that can better model the global variation. So for instance, here, in addition to the usual reference, you have 3 other haplotypes that are ordered from 1 to n that exhibits all the alleles in the sample we want to genotype. While the first, third, and fifth block here are shared, so those that are highlighted in light gray, let's say. The red allele, the blue and the yellow one are present only in certain haplotypes. Yeah, and reads here can map without ambiguity. Of course, so the effort of generating these high quality assemblies that then can be included in larger reference panels is the aim of several consortia, including the AGSBC, which is the Human Genome Structural Variant Consortium, and the HPRC, which is the Human Pangenome Reference Consortium. So AGSBC latest release contains like 65 deployed telomere-to-telomere samples, while HPRC released 47 assembled individuals in the year 1 release, and now there are more than 200 individuals. So generating these high-quality assemblies is somehow still not that cost effective. So that's part of the reason why still linear reference-based approaches are used. And we as Technopol have a partnership with the HPRC, and we have just finished the sequencing and the assembly of 4 TSI individuals, which are basically people from Tuscany, and those will be included as an Italian representation in the next Pangenome release. So we are quite proud of that. Okay, so there are a couple of methods out there that can be used to genotype individuals, so solve the problem that we presented now using a collection of assemblies, for instance, in the form of a pangenome graph. I won't go through all of them, of course, so I talk about 2. So the first one is Pangini from Jana Ebler and the Masha group. And the other one is COSICT, which we developed at the Technopole together with Eric Garrison and Peter Studman's group. Of course, since I'm not the developer of Pangini itself, I may miss some bits, but I find the approach really interesting, and that's why I'm bringing this here. I mean, again, for like didactic purposes. Okay, that's the paper. So everything I will tell you about this method we also find in the paper. I've just tried to summarize it like with sketches so that it's easier for everyone to remember. Yeah? So Pangini relies on a pangenome built from a collection of variants in BCF format. That's one way to represent Pangenome. So I'm not sure if you already went through methods for Pangenome construction, probably yes, but it is just one way to do that. So in particular, in the paper, they used haplotype resolved assemblies for 14 samples. So those, for instance, that you see here. In practice, for each sample, you can separately map contigs of each haplotype to the reference genome. So in that paper, they use GRCH38. So what you see here with R. This can be done using standard aligners, such as Minimac2, and then they covariance. Yeah? So you have the first haplotype, you align against the reference, and then you do variant coding. Yeah? And for instance, here you see that the haplotype 1 and reference differs for a certain block, which is the green one here, which is red in the reference. You do the same for the second haplotype, and you see an additional bit of variation. And then again, so on, you do the same until you align all the haplotypes in the collection. Yeah, and then you can use these variants to construct the graph. In particular, the graph they construct is acyclic and directed, and it is constructed by progressively inserting the variants of all haplotypes into the linear reference genome. So basically, you start by inserting the green block here, and you generate this bubble. Then you add the yellow part, which results in an additional bubble, and then you add variants from the HN haplotype, which result again in a more complex bubble. Yeah? So variants produce bubbles in the graph with branches that define the alleles. The haplotypes, of course, are passed through the resulting pangenome. Yeah. So what then Pangini does? So for each bubble, it determines a set of k-mes that uniquely characterize the bubble itself. So this is done by counting all the k-mes along the haplotype paths and determining then a set of k-mes for each bubble that occurs at most once within the variant bubble itself. So that's for just an example. So you will have, for instance, 4 unique k-mes for the green part of the bubble, 3 for the light blue one, 2 for the red, and so on and so forth. Okay, so once we have the setting here, so we have the k-mes from each bubble, and then you get a sample that you want to sequence from which you get short reads. In practice, what they do, so they count the k-mes from unique bubbles into the sample reads and integrate this in a more complex model that we will see in the next slide. So basically, you have the k-mes from unique from unique branches of the bubbles, and you count this in the reads. So for instance, this k-mes here is seen in 9 reads. This other k-mes here is not seen at all, and this makes sense because this part is not present in this specific sample here. Again, for the yellow, this is seen in 4 reads, and for the dark blue one, so you see seen in 3 reads. From this, you can derive copy number state. So very naively, this can be done by just dividing the number of reads that you get by the average number of reads for each k-met that you see. So basically here it will normalize to 2, to 1, and to 1 here, and 0 because this is not seen, for instance. Okay. So the distance, so the number of k-mets that you see and the distance between bubbles, those are informations that are integrated into a model, which is an HMM, and these like models the unknown genome as a mosaic of the provided haplotypes, yeah, and reconstructed based on the k-met counts observed in the sample sequencing reads. So because you observe these counts here, the genotype will of course be green for the first block and yellow-blue for the second block. And the distance, as I was mentioning before, between bubbles is also taken into account in the hidden Markov model. That is, it somehow influences the transition probabilities of the model itself. And this is also, of course, useful to genotype bubbles that exhibit some uncertainty. For instance, here, in the first bubble, k-met count suggests that the sample probably carries the alleles of the green and the blue haplotypes, while in the second bubble, because this is poorly covered by k-mets, you may have some uncertainty. But because this bubble here is linked to the previous bubble, this can be used to infer the two local haplotypes path for the second bubble, yeah? And it will likely result in the green-blue path inferred from the first the previous bubble genotype, yeah? Which is, yeah, I mean, I think kind of a pretty cool approach. It has some limitation, of course, especially in scalability, but the interesting part, I think, it's the approach itself. Yeah? There is another tool, which is Lossityper from the same group. I'm not going into details of this as well. I'm just leaving this here in case anyone wants to give it a read. So in Pangini, they use a sort of augmented sequence graph model. So you start from the reference and progressively add variation. Yeah. So we have developed our own method of genotyping from graph. This is called COSIC, that stands for cosine similarity based genotype. In COSIC, we use the variation graph model, which describes the all-to-all alignment of many sequences as works to a graph whose nodes are labeled with DNA sequences. So let's just try and build a graph from these three samples here. So we have 6 sequences, right? So in this, can be considered a pangenome itself, because pangenomes are basically just a collection of sequences, but we can use these sequences to build a variation graph. So the first block, which is the black block, is shared across all the haplotypes, and you will have like a node, and then from the black one you can go through to the red or to the green node. So you will have either, so you can take like the red path or directly the green one, or go through the red one and then to the green, and then you have blue and purple, or I don't know what kind of color is this, but there's also one haplotype that loops twice over the blue node. So this is why you see a double loop over this node. And then again, you can go through the yellow or the pink node, and then again to the gray one. That's how you construct the variation graph from these 6 paths. Yeah, and of course those are nodes. So those here are represented one linked to the others, but of course those are nodes, and there are edges connecting nodes. So if you take, there are a couple of concepts that are useful to understand the approach that we use. So the first one is defining the so-called path coverage vector. So if you take the node from the graph and you like plot like this, there's just like a collection of all the possible nodes in the graph, and then you can count the number of times each path crosses each node in the graph. So for instance, for these two paths here, you will have something like this. So the first path crosses the black node once, the red node once, again once for the green node, once for the blue node, once for the like pinkish node, once for the yellow, 0 for the pink, and 1 for the just because the pink node is not present in the path. Yeah, and you can do the same for the second haplotype. So you will have coverage 1 for the black node, 0 for the red, 0 for the yellow, and 1 for all the others. So this is the so-called path coverage vector. So the number of time each path crosses a certain node in the graph. And it's node space because it takes into account all the possible nodes, because of course some paths may not have certain nodes. Once you have a sample, for instance, those are reads that are generated from a sample, you can take the reads and align those reads to the nodes in the graph, because nodes are sequences, so you can just take the reads and align to the nodes and derive a sample coverage vector, which is just the number of reads aligning to each node in the graph itself. And that's exactly what I was referring to. So for instance, you have 24 reads here, 12 reads in the red node, and so on and so forth. For all the others, you see numbers here. Okay, and these three concepts here, so the variation graph, the path coverage vector, and the sample coverage vector are everything that we need to try and genotype a sequence sample using a Pangenome graph, using the approach that we built. So that's the workflow. The thing that you see here is on the left is the so-called rule graph. So the rule graph is like tells you where you come from, where you go when you specify a certain rule, and this comes directly out from one of these 2-3 workflow languages, which is SnakeMake, and indeed the workflow itself is implemented in SnakeMake. We are going to break this workflow down into pieces, so don't worry if it looks like confusing at first. So let's say that we want to genotype a certain region in an individual. What we do can be summarized more or less in 4 steps. So the first step is to build a graph for the region. So that's a region-specific graph, because working with graphs that are too large becomes impractical. So the first stuff that we do is to pinpoint to the region we are interested in, and then we build locally a variation graph for that part. And then we compute the sample coverage, what I was mentioning before in the introduction. We compute the path coverage, again, one of the previous slides. And then we do the so-called haplotype deconvolution, which is the genotyping itself. So at the moment, our workflow requires as input, so chromosome level assemblies like those that are generated from like HESVC, HPRC, or whatever, just those that we know like belong to a certain chromosome, reads from one or more individual and one or more region of interest that we want to genotype. And the output are like the sample haplotypes, so the 2 haplotypes in the graph that best describe the sample, and the structural clusters for these haplotypes. But yeah, we will see this like in a couple of minutes. So the cool thing of having everything implemented in a workflow language is that it's easy to genotype in parallel. That is, we can like do what I'm describing now for a single region across many regions and many samples at the same time, which is pretty convenient if we want to scale at biobank scale or like many samples, yeah? And many regions. Okay, so that's the, as I said, we have like four main steps, which are those that I'm going to like explain a bit better. And so what you see here in gray and black is the reference, and we refer to these as target. And the other assemblies, we usually call them queries. Yeah. So the first thing that we do is a query to target alignment. And then we leverage the so-called implicit pangenome graph model to extract the region of interest. So basically, we have the queries aligned to the target, and we can use the implicit pangenome graph leverages COI trees, which are implicit interval trees, to provide like efficient range lookup over the input alignment. So we have the queries aligned to target. We know the coordinate of the region of interest in the target. For instance, a certain region of GRCH38. We can use the pangenome, the implicit pangenome graph model, and the COI trees to project the region of interest from the target into the queries. Yeah. So once we have the queries aligned to the target, so we project the region of interest into all the assemblies. And once we have the coordinates of the region of interest on all the other assemblies, we can just pick these regions from the assemblies and use them to build a variation graph. This is done using PGGB that has been developed by Garrison and Guadrascino. But yeah, in effect, and this allows us to build a region-specific graph. And for instance, here you have the C4, which is the complement for region on human chromosome 6, which has these aspects here. This is just a sketch, of course. The true aspect is like this one here that you see on the left. So once we have the region specific graph, we can compute the path coverage, and we said that the path coverage is just the number of time each path crosses a node in the graph. So what you see here is basically the path coverage for two different paths. So the first path is haplotype 1 from this individual here, AG00096, and the graph that you see, the plot that you see in the bottom part of the slide is the second haplotype of the same individual, right? So each point here is a node. What you see on the x-axis is the cumulative length of all the nodes that are in the graph, and you see that some nodes have coverage 2, which means that path crosses that node twice. Some others have 1, and some others have 0 because those nodes are not represented in that path. That's the same for the second haplotype of the AG00096 sample. So some nodes have 1, some nodes have 0. You don't have nodes with 2 coverage, for instance, here. Just another example for another sample, this AG00171. So you see that some nodes have 0, some nodes have 1, 2, and there are just a couple of nodes that coverage 3 and 4. Yeah, which basically means that that path crosses that node 4 times or 3 times with respect to the height of the point that you see here in the plot. And same here for the second haplotype. Yeah. Okay, so because we want to genotype a certain sample, so we take his weights and we map them to the regions that we have extracted from the assemblies. And this can be done using standard aligners. We use VWA-MEM for this. We use actually VWA-MEM2, which is like a bit more fast. But in practice, you can ideally think about extending this to minimap2 with long width presetting if your input are reads coming from Oxford Nanotechnology sequences, for instance, or PacBio sequences. And once we have the reads mapped to the blocks that we have extracted from assemblies, we can project back the coverage that we have for each block into the graph. And what we have, it's a sample coverage vector. So if you just look at the upper plot, each point again here is a node, and if you look at what you have on the y-axis on the left, yeah, that's the sample coverage. So for instance, here the sample has coverage 100, here sample has coverage 40, more or less. And this can be overlapped with the pangenome coverage vector. So for instance, here you have reads coming from AG0096 sample that's been sequenced with short reads, and below you have the pangenome coverage for the two parts of the same individual overlapped with a sample coverage. Yeah? So the two y-axis have different scales, because of course one is from the sample and the other one is from the pangenome, right? You can of course overlap these to other haplotypes. So this is, for instance, the same sample, but the pangenome coverage represented here comes from haplotypes coming from a different sample, so AG000171. So once we have this setting here, so we have the part coverage vector and we have the sample coverage vector. So the question that we want to address is the haplotype deconvolution itself, which is the proper genotyping step. So the haplotype deconvolution problem can be break down to find the combination of parts best representing the trend observed in the coverage vector. And of course, the combination will be of two parts, because we are dealing with diploid samples for human, and each part is haploid because it's like a single haplotype, and we have two, so we need to combine two parts in the graph and check which is the combination that best fits with the sample coverage vector. And that's why n equal to here for a diploid sample. What we do in practice is to compute. So this can be done in many different ways. We chose to use the cosine similarity for that. So the cosine similarity is basically the angle between two vectors is calculated. So having two vectors is calculated as the dot product divided by their norm. And in practice, what we do is schematize as follows. So we have the sample coverage and we have the part coverage vectors. So what we have highlighted in the previous slides. We generate all the combination of part coverage vector of two. This includes, of course, for heterozygous and homozygous combinations. So you will have for these three H1 plus H2, H1 plus H3, H2 plus H3, but also H1 plus H1, H2 plus H2, and so on and so forth. And then we calculate the cosine similarity of the sample with respect to all the possible haplotype combinations. And then we pick the combination with the highest cosine similarity score. So for instance, here, this sample coverage vector is best described by the combination of H1 and H2, because the cosine similarity here is the highest. Yeah. One thing that is actually true is that many haplotypes are structurally similar, and that's something that you can also see by eye. So in this plot, actually the haplotypes are already sorted by similarity, but each line here is an haplotype. And what you see is that many haplotypes actually are very, very similar. For instance, you have a first block, let's say this is group 1, with haplotypes that they look like very similar, right? Because the red part is where the part coverage increases. So you will have some regions that are gray, which means that they have 1 in terms of part coverage, and then you have some regions that are red. which means that they have 2 or more in terms of path coverage for the nodes that are highlighted in this region. Yeah? And then you have another group again, so group 1, group 3, group 2, group 3. So all these haplotypes here, in terms of large variations, are very similar. Yeah? And then you can define at the same time group 4, group 5, group 6, and group 7. So what we found was that it kind of makes sense not to just output the best combination of haplotypes that describe a certain sample, but also define the structural cluster of each haplotype, so that we can predict not just a single combination, but the haplotype cluster each haplotype belongs to. At the moment, we are doing this using just agglomerative clustering with a silhouette analysis for defining the best number of clusters, and then once we have the number of clusters, we cut the dendrogram tree, and we use that many clusters that is it comes out from the silhouette analysis. But in practice, this makes a lot of sense also from a gene structure point of view. So what you see here are clusters with gene injected, like those are gene annotations that are injected in the graph, and haplotypes from the same group here are very similar structures. For instance, all haplotypes in group 2 here miss, for instance, C4a. So the region here is the C4 on chromosome 6, which is characterized by C4a, C4b, and some other genes that are not that important at the moment. But all haplotypes, for instance, in group 2 miss C4a, but also some other bits, while those in group 3 have, for instance, longer C4b with respect to those in group 1. That's just an example. By like predicting a couple of haplotypes, maybe like you may be able to predict a couple of haplotypes that falls in the proper cluster without predicting the proper one. So let's say that we have a sample like it's AG or 96 or whatever. We may not be able to predict exactly haplotype 1 and haplotype 2 from AG or 96, but we are still able to predict clusters from the proper clusters from the respective haplotypes. Yeah. So that's just an example of benchmarking that we do. So we have HESVC version 3, for instance, 65 samples. From these 65 samples, we have 64 matching short reads that are coming from 1,000 genome. So we take a sample, we genotype. So we take a sample that has been sequenced, we genotype with respect to the graph, and we assign both the couple of haplotypes and the structural cluster to each individual, right? And then how do we check if our predictions are correct? So we ask basically 2 questions. So if the sample has been assigned the proper haplotypes, we expect it to be S1, S2, because they are in the graph, so we are able to see if those are like correct. Or if we have a sample that is being assigned the proper clusters for the two haplotypes. Yeah? In that case, it would be a true positive. Otherwise, it's an error. And for this region here, we have 63 out of 64 of these samples that are properly genotyped. So this basically means that our accuracy is pretty high, at least for this region here. Of course, we can repeat this by like removing one haplotype from the original graph and try to like regenotype the sample, checking just for cluster, and this will be called leave one out. But yeah, we can talk about this later. I mean, just in the interest of time, I'm gonna skip this part. Yeah, okay, so that's more or less how it works. We are like currently experimenting with like refining this approach. So one thing we want to like do is to explore if like removing nodes that have stable coverage across all the paths somehow helps, like improving the genotyping itself and the clustering, because of course the nodes that have stable coverage across all the paths do not contribute to variation, and therefore should not be like have a big weight like when clustering, right? And then another thing that we are doing is like not using agglomerative clustering, but instead switching to dB scan so that we can better control the intra-cluster similarity of the elements that goes within each haplotype cluster. And at the moment, we are also extending this to many other loci, and the idea will be to run this across at least 100, 200 loci before having a stable release that we can publish. But okay, so of course this is not just work of the Technopole, but we have many collaborations with people, especially in US. Eric Garrison and Andrea Guarracino here, and Peter Sutman and the group is Jason Chin that really helped us a lot, especially in the first part of the development. So this approach has already been published in Nature as part of this paper here that's focused on the amylase. So basically what I showed for C4 locus, this has been applied to amylase. What you see here on the right are the structural clusters of amylase as they were defined by the approach that are described before. And I'm also leaving this here if you have like if you want to give it a read or like curious about how this can be applied to different data sets, because here we didn't just use it on modern short-read samples, but also on ancient DNA samples, and it works pretty well also at very low coverage levels. Yeah, and that's it from my side.
Thank you very much. It was very clear. And we are waiting for questions from the audience. Again, I remind everyone to write your questions on the WhatsApp channel. One quick question from me. What are the computational requirements? So how long did it take? Because I mean, it's impressive what you have done, but I want to see how easy it is to replicate.
Yeah, okay. So I guess it really so this is like more convenient to run if you have many regions that fall within the same chromosome, right? Because let's say that you want to genotype 100 regions that are on chromosome 6. So the very first part, so the alignment where you have all the queries to target alignment is done just once per chromosome. So let's say that you have all the contigs from chromosome 6. So you align all the queries to chromosome 6. And then once you have that part, right, it's pretty fast. Because so once you have the alignment of all the queries to the chromosome, then like slicing and identifying the single blocks inside the chromosome is super fast. And also the local graph construction, which otherwise will take like days, right? Because building a graph for an entire chromosome is too time consuming. But because we are doing this locally and in parallel, then it's super fast. So for instance, like I don't know if you have for amylase locus, just one locus, but we genotype this across thousands of samples, and it took like 3 or 4 hours. Yeah, so that's
3 or 4 hours, but on a huge server.
It's just it's just like one computation. Yeah, it's like 32 nodes, but just each node using one CPU. So it's like, well, you can't actually think of parallelizing this without efficiently parallelizing this without having a cluster. That's you need to rely more or less on a cluster for efficient parallelization. But yeah, that's what I mean. I mean, if you have like some computational power, like 32 CPUs, for instance, then it's like super fast. So the efficient, the part that's more that takes the more time is, as I was mentioning here, the alignment of queries to targets, which is like a bit time consuming. It's like alignment, but all the other stuff, it's very local, so it's like reduced to the minimum terms.
And is the software available?
Yeah, exactly. So I think I have like the link here. Yes, exactly. So it's
okay.
Yeah, it's also present in the slide.
Okay, let's see. So I see no questions in the chat. Let's wait a few seconds if someone asks there. Otherwise, I think that we can thank again Davide. A very, very nice talk. I enjoyed it a lot. And thanks again for being here.
Thank
So
you.
it was a pleasure. Goodbye to everyone. And so we are going to resume the session at 2 p.m., which is slightly more than 2 hours from now. So see you everyone there.
Ok, mi sembra andato bene, lo streaming è
Ok.
interrotto.